Hi everybody, my name is Lauren Salminen and I am the chair of the ENIGMA-Environment Working Group. I put together this video to describe kind of the general goals of ENIGMA-Environment according to what our existing NIH grant funds us to do, as well as data sharing principles and what our actual process is. We presented this at the ENIGMA leadership meeting in Amsterdam back in December, and we received a lot of really great feedback from groups that were interested in participating, but had a lot of concerns about data sharing, especially for those in Europe, given the data sharing policies related to GDPR. So I just want to spend a little bit of time going through that, and hopefully if anybody has any questions, some of this will clear it up, and please feel free to reach out. And we're happy to set up individual meetings to figure out what might work best for your group. So I lead this group with two other colleagues at USC, Megan Herding and JC Chen. They are faculty in the Population Public Health Sciences Department. And then we have the wonderful Dr. Teresa Monreal, who is our project manager, and she's really responsible for coordinating data flow, communication with each of the cohorts, and just general organization. So we have complementary areas of expertise. My area focuses on brain aging and risk for Alzheimer's disease and cerebrovascular disease. Megan's work focuses on development, and JC is a epidemiologist and toxicologist with an emphasis in brain aging and Alzheimer's disease. So our current grant focuses specifically on air pollution. We are ENIGMA-Environment, and so the goal is really just to understand how various elements in the built environment influence brain health. Right now we're focusing on air pollution, given that NIH has been increasingly concerned about the various health consequences of ambient air pollution. So our existing grant focuses on two primary pollutants, nitrogen dioxide or NO2, and fine particulate matter, which is PM 2.5. These are two of the 6 criteria pollutants that were identified by the United States as pollutants of concern that needed to be monitored. PM 2.5 is the most commonly studied, and that's because it has known to have the most harmful health outcomes, and that's due to its microscopic size. So in the slide here, you can see PM 2.5 relative to a strand of human hair, and that's those little tiny pink beads on the blue beads, which is PM 10. So essentially it just refers to the size of the particle. And we know from various different studies that the smaller the size of the particle, the worse it is for health outcomes. And that's because it can penetrate different biological barriers. So PM 2.5, meaning that the max diameter is 2.5 microns, can embed deep into the lung alveoli and translocate into the bloodstream and pass the blood-brain barrier to access the brain. There's also compelling evidence that it can directly access the brain through nose breathing and can pass over the olfactory bulb. Part of this is because ultrafine particles actually dominate PM 2.5. So there's a lot of reason why we would want to look at this in relation to brain health across the entire lifespan. One of the limitations, it's a fairly significant limitation of existing literature. There's not that many studies that have looked at neuroimaging in relation to air pollution, but of those that do, and many of those focus on PM 2.5. The problem is that, again, it just refers to the size of the particle. So we're not getting any information about the underlying chemical composition. And we know from existing work, and this slide represents that, that this is a big issue because the chemical composition varies dramatically by geographic region. So PM 2.5 in Los Angeles, where I am, is not going to be the same as PM 2.5 in New York or in South Africa or in Europe. Within Europe, there's going to be different concentrations of these. And the chemical components that are listed around the total PM 2.5 mass on this slide are the chemical components that can be reliably extracted on a global scale. And there's been a lot of studies to show this. And so one of the goals of our existing R01 is to understand how the underlying chemical composition relates to brain integrity. And the reason for that is because although most studies show an adverse effect of PM 2.5 on the brain, there is quite a bit of variability from study to study in terms of the specificity of those effects. And that is likely due, at least in some part, to the underlying chemical components. So Dr. Randall Martin is a chemical transport scientist at WashU in St. Louis. He is our collaborator who has really spent his career developing and validating these measures of atmospheric particles. And he developed GeoSchem, which is this very elaborate system to leverage satellite observations of atmospheric components. So essentially what GeoSchem does is that it calculates the temporal and spatial evolution of aerosol and gas compounds using meteorological data sets, emission inventories, and various equations that represent the physics and chemistry of these constituents. So Dr. Martin has already shared all of the pollutant information with us that are basically on 1 by 1 kilometer grids. Those have been shared with our USC team so that those estimates can be linked to residential location. And here is where the privacy issue comes into play. So we have a team at USC, Dr. John Wilson and Bo McDonald, his colleague, they're at the Spatial Sciences Institute, and they are our geocoding personnel. They are responsible for doing, for transposing residential address information to geocodes. If they don't already exist, some cohorts do already have their addresses geocoded. And then for all groups, even those that might already have air pollution information, which there are a few, they are assigning the air pollution concentrations based on specific timescales to those geocodes. This way it's done in a systematic way for all of the different participating data sets. So we're doing this in a centralized way because there is no universal format for address inventory. So we couldn't just put together a script and just send it out so that groups could do their own geocoding. And that's because there's various different geocoders. They don't all use the same reference data set. There's a lot of different elements that I didn't even realize went into the complexity of geocoding. So we're aiming for the most precise estimate of home location as possible. So including a house number, that would be the most ideal. But there is some flexibility in that. So if you have street level addresses, and also does depend geographically where you are, because population density also determines zip level and how much coverage a zip code may provide just based on population density. So for some groups in Europe, they had the street, but they didn't have necessarily a home number. And based on the location of those cohorts, it was fine. So for PM 2.5 in particular, it's a bit more stable over broader distances. So we have confirmation from Dr. Martin that even going up to a kilometer or two would not be an issue at all. Other pollutants like nitrogen dioxide, which is a gas, that's commonly used as a proxy for traffic-related pollution. That is much more sensitive to, it has much more spatial sensitivity. So the further you get away from the road, the more that NO2 really just drops off. So that's something that we'll have to consider when we get into the NO2 analyses, but because PM 2.5 is the most commonly studied, and luckily it is more stable, it does give us a little bit of flexibility to include groups that might not have a house number, but they do have the street. And so we can geocode from a centroid at that point. So the way that we do this is we have a two-pronged approach. And what do I mean by that? So each cohort basically puts together a Excel file with very little information. So on the slide, you see there's a column for ID, there's a column for the MRI scan date, the date at which the residential address information was collected. Oftentimes that's not at all, or maybe even close to the MRI scan date, but we do need to know what that kind of relationship is since we're linking these measures to brain metrics. And then the address. And a lot of cohorts, honestly, that have shared their data, they're even within a cohort, there might be some variability. Some participants may have a house number and some might not. That's whatever is recorded is, we can work with it. So there's complete separation from what is sent to John and Bo's team, then the central analytic team, which is myself, Dr. Monreal. We have Dr. Jinwei Wang, who's a biostatistician that we're working with. We don't ever see the file that is sent from the local cohort to the Spatial Sciences Institute. And that is really to have that complete protection so that later when we do receive the actual covariate file that has clinical information, demographic information, we aren't able to go back then and link that to address because we've never seen that file. And because the file that's sent to the Spatial Sciences Institute only includes these 4 columns, there's no PHI because literally there's no health or demographic information. And we also have had several groups dummy code their IDs so that the ID that is sent in the file that's sent to the Spatial Sciences Institute is not the same as the subject ID that's sent in the main covariate file. So, the first step is that the cohort shares the address and geocoded data. Once the address has been geocoded and the exposures have been mapped, Bo and John return those data to the local cohort. The actual outputs are also saved so that we can do any cross-checking, but the geocodes and the address information is never shared. It's a very secure file transfer process that is set up through a HIPAA-compliant computing environment at the Spatial Sciences Institute. This is what they do all day, is essentially geocoding and mapping of various environmental measures for various different data sets. So they're very experienced in doing this kind of work. The exposures are returned to the cohort, and then the cohort takes the outputs, the air pollution outputs, and puts that into their covariate file, and we have a specific covariate template with the variables of interest, and only that information goes in that file. So they're certainly not including their address information or their geocodes in that main file that's shared. So then after that, the exposure outputs are shared. The main data file is shared after the geo-linked exposures have been returned to the cohort. So this is going to be the file that's actually analyzed. So the most important takeaway from this slide, even though there's lots of parts, is that the Spatial Sciences Institute, Bo and John, never have access to protected health information. In the same way that our central team never has access to geocodes or addresses. They're completely separate processes. And we specifically set this up in this way so that there could be that complete separation. We had several meetings with our IRB to make sure that this would be acceptable. And we actually have not had, to my knowledge, we have had several European cohorts share their data, and we have not had any issues. The other option that we have for cohorts that have very strict data sharing policies is geocode blurring. So this would be for cohorts that already have their data geocoded. Any cohort that does knows that you can reverse geocode to find an address. So it's not like it's some encrypted information. It represents an actual address. So we have an R script that we can send to that cohort, which can jitter essentially the X and Y, the latitude and longitude coordinates. And it blurs it up to 500-meter radius. And we can account for that in our statistical models. It will not have any meaningful impact on the outcomes. And this was according to several conversations we had with Dr. Martin and the geospatial team. So we have had a couple cohorts use this option so that just for an extra level of security, even though there's no health information that's shared along with the address or the geocodes, it just provides that extra layer of protection. And then the last option that we have used for at least one cohort is remote geocoding. Bo McDonald, she's primarily responsible for doing all of the direct work with each data set. And they have software, this AnyDesk software, where essentially we can remote log into the local cohort's computer with their permission and all of the necessary elements set up. And we have all of the instructions in this USC letter that really outlines all of the steps that need to be taken. We've done this before, it has worked successfully. So this is really for cohorts that do not are unable to or feel uncomfortable with sharing any elements, having them leave their local computer. This way, nothing would leave the local computer. It would stay put. So for these three different options, this really kind of gives us the most coverage, the most flexibility to work with each individual cohort. And again, there's not any cohort that we've presented these options to that have not been able to share, including those in Europe. So that's been really exciting. And hopefully this cleared up some of the data sharing concerns that I know a lot of groups have had, even though they've been interested in participating. And again, we're happy to meet with individual groups if there are still questions here. This is really kind of the main point of emphasis, is just so that people know that they're never sharing any protected health information. There's complete separation from the different teams that will be handling a cohort's data. And we do have a lot of these safeguards in place for extra levels of security. So please let me know if there is anything else I can clarify, and hopefully this maybe address some concerns and we can continue to grow and build our data set within ENIGMA-Environment. We've already begun analyses and are starting to progress towards our first paper. We have one abstract that was accepted for a symposium presentation at Society of Biological Psychiatry, and we'll be presenting that in Toronto in April. We also submitted an abstract to the Alzheimer's Association conference, and we anticipate a lot more exciting projects to come. So please reach out if you have questions, and we hope to hear from you soon. Thank you.